Yeah. And I welcome the organizing committee members as well. From JC Bikaner, we have Dr. Jakhar, the techie coordinator. We have Dr. Praveen. Dr. Jakhar, maybe you can wave once so that Madam gets to know.
Good afternoon, madam. Good afternoon.
Techie coordinator. And Dr.
Good afternoon.
And Dr. Praveen, he is head of the Department of Civil Engineering, otherwise is in chemistry. Then we have Dr. Prashant Bhakad, I think if he's there. So the one in specs is Dr. Praveen, and then we have Mr. Ravi looking after as the host of this session. And from NIT side, we had Dr. S.D. Chalak, he is faculty in Civil Engineering Department, and another coordinator, Dr. Professor Surendra Biswal. He is professor in civil engineering and otherwise looking after as registrar of NIT, registrar in charge. So that's how the team comprises. And anyway, I'm there as a thread in between, connecting all these people and trying managing things, just kind of helping them out. So with that note, I welcome you all once again. And I invite Dr. Jakhar to formally welcome the honorable speaker and all participants and say a few words. And Dr. Jakhar, please.
Thank you, sir. Thank you. Good afternoon, everyone. I feel immense pleasure to formally welcome Professor Sudha Goel as a speaker for this session, and I hope her talk will be very fruitful and beneficial for the participants. And I also welcome all the participants in this session. Thank you so much, and over to Professor Satha.
Thank you, Dr. Jakhar, for extending a warm welcome to the speaker and to all the participants. Now I go on to Dr. Praveen Purohit to please do the honor of introducing the speaker to the participants and to us all. Dr. Praveen, please.
Thank you, sir. Thank you very much for giving me this opportunity to introduce Dr. Sudha Goel to this.
Dr. Praveen, keep your mic a little closer to your mouth. Yeah, please.
Sir, now am I audible?
Yeah.
So, a brief introduction of Dr. Sudha Goel, and she is Associate Professor from Civil Engineering Department, IIT Kharagpur, and she has a tremendous academic record.
Dr. Praveen, your voice is a little low still. I don't know what is the reason. Maybe check your volume of your laptop. On the bottom taskbar, put it on the maximum, and then keep the mic a little closer. Maybe that will work.
So now is it okay?
It's better, maybe. Yeah, please go ahead.
Yeah, she has done her PhD from the Johns Hopkins University, Belton, USA, and her MSc from the Johns Hopkins University, Baltimore, in May 1992. In June 1990, from College of Engineering, Gujarat University, LD College of Engineering, and she has done her PhD thesis under advisor Professor E.J. Browser, GHU. Now, she has been awarded by a number of awards. She has got in 2015 the Best Paper Award, and 2014 the Wiley Discovery Research Officer, the Online Quiz Award, and in 2010 Professor R.C. Singh Award, and in 1996 as the Best Student Paper Award. 1990 PP Oza's Gold Medal. She has been awarded with this. She has a professional experience. She has Indian Institute of Technology, Kharagpur. She has been working as associate professor, and then she has actually a number of publications. She is the author of many books and etc. And she is actually the area of interest of her research is environmental engineering, the water quality and treatment, solid and hazardous waste management, environmental risk assessment, environmental impact assessment, and she has been awarded with a number of medals and awards there. And she has been associated with a number of projects also. So many of these projects have been with the foreign countries also, and she has been teaching a number of courses like advanced water and wastewater treatment, engineering drawing and computer graphics, and a number of courses in IITs and other. So this was all a brief introduction of ma'am, and we are being happy to have such an eminent speaker, and we will be benefited surely by the topic she has been delivering here. Thank you very much, madam. Now I hand over the session to Professor Sathan. Thank you, sir.
Thank you, sir. Thank you very much. Thank you, Dr. Sudha. Thank you very much for giving me this opportunity to introduce Dr. Sudha Goel to this session. Now I hand over the session to Professor Sathan. Thank you, sir. Satan sir, please carry on.
Okay, Dr. Praveen, anyway, I could somehow make out, what you were saying, but then, yes, to the participants, to and everybody, I can say, she is a very versatile person, having international exposure and experience, done her qualification from very wonderful universities in USA, and all in all, into so much good research into R&D projects. So in the nutshell, I can say a very, very accomplished person, very accomplished faculty and a researcher. So this session, I can say to the participants that they can look forward to a really wonderful talk. So thank you so much, madam, and thank you, Dr. Praveen, for introducing the speaker to us. So, madam, I think without taking much time, I'll come over to you. You may now start your session, please. And you're welcome once again. Yes, yeah, please.
Good afternoon, everyone, and thank you so much for introducing me with such compliments and so on. I don't feel like I deserve it, but thank you so much. Can you hear me clearly?
Yes, madam. Yes, we can hear you clearly. Absolutely fine.
You can hear me. Okay.
Yeah.
So then what I will do is I will start sharing the content, the slides that I have.
Yeah.
And let me, yeah.
And participants, meanwhile, in case there's any question, they can keep posting in the chat box. And then maybe we'll take up all your questions with the speaker at the end. Or in between also, in case there is very pressing question, maybe you can interrupt. Otherwise, you can keep posting your question in the chat box, right? And at the end of the talk, we'll take up all questions, and maybe you can ask questions directly also at the end. Madam, please go ahead.
So I hope everyone can see the title slide.
Yes, ma'am, it's visible.
Title slide is visible. Okay. No time lag in my talking and your ability to see the slide and so on, right? Okay. Yeah. All right. So, oops, I lost that. Okay. So my talk is going to be about xenobiotics and bioremediation. And like I said, I'm very happy to be part of this course and give a lecture on what is perhaps a new topic. It's been around for almost 50, 60 years, but I don't see much activity. There is some activity in India about this, but not too much. So maybe in the future, we'll be seeing a lot more of this. Many of you might be wondering what is xenobiotics. So,
Madam, you need to share your slides again. You need to present your slides. The slides are not visible, so you need to present it again, maybe. And Ravi, you might have made Madam as a presenter, na?
Yes, sir. Yes, sir.
एक बार आया था मैडम, but slide चला गया. हाँ, I think yeah, it will come now. Please put it on the full screen, ma'am, so it's visible, right? Perfectly fine, ma'am. Please go ahead.
Okay, thank you. Okay, so I'm going to be talking about xenobiotics and bioremediation. And even though the idea of xenobiotics is as old as, it basically started in World War II, and it's been going on since then. And so is bioremediation and so on. So it's been around for a while, but in India, we haven't seen too much activity in this area. There is some activity, but not a lot. So hopefully we'll have some, what I'm going to do is, I'm going to go through some of the fundamental concepts that are applicable to this particular topic. So for those of you who are not familiar with the idea of xenobiotics, xeno is basically alien or foreign. In this case, what we mean is foreign to the environment. And biotic is anything that's biologically active. Or in this particular case, because these compounds are considered to be alien or foreign to the environment, their biological activity is somewhat different from what you would expect for a natural organic compound. So the most common examples are synthetic organic chemicals. Synthetic organic chemicals, like you all know, are pesticides, herbicides, any of these chemicals that we use for controlling the presence and reproduction of pests, herbs, weeds, all these kinds of chemicals that we buy from the market and use in our gardens, farms, and so on. Then there are two other groups that have recently been included in this definition. The first one I've already mentioned. The second one is natural compounds. Now, if you have natural compounds that are present in higher than what is considered usual or background concentration, then that is also considered to be a xenobiotic these days. And the best examples are hormones. You may have seen research papers, you may have seen media reports about extremely high levels of estrogens and so on in streams and wastewaters and their impact on the reproduction of various organisms. And then we have a third group. This third group is any chemical that's novel to the metabolism of an organism, including human beings. So any synthetic organic compound or any other synthetic material that is non-biodegradable, plastics, dioxins, disinfection byproducts, these are all examples of compounds that are formed by anthropogenic processes, and they may be novel to the metabolism of an organism. For example, we do not have the ability to digest plastics. If you have small amounts of plastic in any material that you ingest, it's normally going to be excreted, but we do wonder what the other problems that may be there associated with such compounds. So that brings us to the question that I have over here in the slide. Why are these compounds a problem? These compounds are persistent. The first thing that they are really am I able to show you the slides?
Yeah, yes, ma'am.
Yeah, we can see, madam.
Okay. Because my screen is not. Okay, so there's no problem in seeing the screen, right?
Yeah, we can see clearly.
Okay. So when it comes to what is the problem with these compounds, the first thing is the word persistence. These compounds are not easily biodegradable. So the first thing is, because they are alien to the environment, they are very difficult for the natural biota, which is not familiar with this particular compound or any group of compounds. Because they are unfamiliar with this particular group of compounds, these compounds will tend to persist in the environment. And because they are persistent, which means they will last for months, years, and so on. Because of this persistence, they will bioaccumulate or bioconcentrate or biomagnify. Now, these are three words that I'm using over here, and I will be defining them shortly before we go through a few other points. In terms of our understanding of xenobiotics, I always like to have some historical background about things that we are generally talking about and learning. So I thought I'll include this slide, which tells us a little bit about how our understanding of xenobiotics has grown. The most, I should rephrase that, the best documented compound is DDT. DDT has been in use since 1939. It was very effective for controlling malaria and typhus during the Second World War and after that. So it became extremely popular among farmers, among people for controlling these diseases. And the consequences of what I would call the use and abuse of DDT became apparent through the 50s. And then the 60s, it became more and more obvious that this compound was having an effect not just on the pests, but also on human beings and other endangered, like it was causing species to become endangered and so on. So we'll go through some of these issues later. In 1961 to 1971, there was another herbicide that started becoming very popular. And I shouldn't use the word popular. It was a wartime use of a particular compound. It's Agent Orange, which was a mixture of two compounds, 2,4-D and another compound. It was very commonly used in the Vietnam War. And because it's a defoliant means it was destroying the canopy, the vegetation canopy, so that the people who were hiding in the forest could be exposed and so on. Now, the consequence of that was that these compounds, which were part of the herbicide, were extremely toxic to those exposed. And the sad fact over here is that people who were utilizing these compounds, as well as the ones that were the victims or on the receiving end, both sides were equally hurt, you might say, by the use of this particular compound or mixture of compounds. Then came the publication of Silent Spring in 1962, and there were several other books by Rachel Carson. She was a marine biologist, and she documented the short-term and long-term toxicity of synthetic organic chemicals such as pesticides. And DDT is one of her big things, but she also talks about various pesticides, parathion, malathion. She has covered almost many of these pesticides. And she was relying on media reports. She was relying on medical reports. All kinds of sources of data were there that she was relying on to say that these compounds were having a very serious effect on human beings as well as on other organisms like birds, fish, and so on. So because of the evidence that was accumulating through the 50s and 60s, in 1972, there was a ban on DDT and several other pesticides. Because by then it was clear that there were developmental and reproductive effects on fish, birds, and several other types of organisms. Now, about DDT in particular, people have found that DDT is the only compound that that mosquitoes are still sensitive to. They've become basically resistant to all other chemical compounds. So WHO is now recommending that DDT can be used with restraint for the use of malaria. I'm sorry, for the control of malaria. Okay, so that is another turnaround, it seems, but you know, it's an interesting story because if you think about how pesticides were manufactured initially, there's been a turnaround on that. And now, if you look at DDT in particular, there's been another turnaround about that as well. So what are xenobiotics? I've already mentioned that they are herbicides, pesticides, and several other compounds come into this group of compounds that are called xenobiotics. Now, anything in the environment can be degraded by physical, chemical, as well as biological reactions. And the same thing is applicable to xenobiotics also. Before we go into the factors that result in the degradation of xenobiotics, let's just take a quick look at pesticide manufacturing. Now, in the late 30s, just around the Second World War, manufacturers were designing these synthetic organic chemicals. These pesticides were being designed in the lab. Remember, they are not natural compounds. They were being literally invented in the laboratory for the purpose of destroying particular organisms, pests that might ruin the crop, mainly crop and agricultural purposes. And they were designed to be persistent. So many of them were chlorinated compounds. These chlorinated compounds have a tendency to persist in the environment for a very long time. They're considered recalcitrant. Recalcitrant means they do not degrade easily. Because of this trait, they tend to bioaccumulate in the tissue of human beings as well as animals, birds, and whatever organisms they may be in the environment. Soon enough, people started realizing that some of these compounds also had what is considered a mutagenic potential. Mutagenic means mutation-causing potential. Now, anything that has a mutation-causing potential, mutagenic potential, is likely under certain conditions to also result in tumors, also result in cancers, and perhaps in reproductive as well as developmental defects. And that is precisely what Rachel Carson had started documenting. And after that, there have been thousands of people who have been working in this area. And it remains an active area of research even today. Once the long-term impacts of these compounds became apparent, so you have these long-term environmental impacts, it became necessary for countries to regulate the manufacturing of these pesticides so that they degrade rapidly. So persistence, which was considered a virtue in the beginning because it made the compound cheaper. So you can imagine that way back in the 40s and in the 50s, farmers would go to buy a can of pesticide, happy to think that, it will last through that season. When it became obvious what the problems are that are associated with these compounds, and the regulations came in that now the manufacturers have to prove to the government that the compound that they have invented and marketed will degrade rapidly. So this is one of the biggest changes in the nature of, pesticides. So the new pesticides are manufactured to degrade rapidly in the environment after they are applied, generally in gardens, fields, and so on. We use a common term, and I think most of you are familiar with this concept of half-life. Now, half-life we generally think about in terms of radioactive decay, but we use the same concept because it comes from first-order reaction kinetics. We use the same concept to basically define the persistence of pesticides in the environment. So if I have compound A, which has a half-life of 1 year, and I compare it to compound B, which has a half-life of 1 month, then that gives me an idea that the one that is persistent for 1 year, the one with a half-life of 1 year, is going to be more persistent in the environment compared to the one which has a half-life of 1 month. Okay, so this is very clear. So we have first order reaction kinetics, and when C is equal to half of C0, that is the half-life. That is the time at which C is equal to half of C0. So you have a reaction or reaction decay constant, and that is basically what we use for comparison. So initially in my first slide, I mentioned 3 words: bioconcentration, bioaccumulation, and biomagnification. There is a 4th term that is mentioned in this table, and that is trophic magnification factor. Now, if you are familiar with biouptake of any particular compound, especially organic compounds, then you're dealing with any or all of these terms. Let's go through some of the basic definitions for each one of them. So we have our bioconcentration factor, which is the ratio of the concentration of that compound in fish tissue divided by the concentration in water. And the assumption is that there is no dietary intake. In the second case, dietary intake is taken into account. So you have bioconcentration versus bioaccumulation. The only difference is that there are several routes of exposure. So you have respiratory exposure, you have dermal exposure, and you have ingestion. Now, out of the 3, in the first case, ingestion is not taken into account. In the second case, ingestion is taken into account. Then we have biomagnification factor. Biomagnification factor is the concentration in fish tissue versus the concentration in the entire diet. Now, people can use, it's very common in this field of research for people to use different organisms. People use insects, certain other tissue can be used. We use bacteria, algae, any of these species can be used depending on the reason for using it. But there are standard assays, standard bioassays in which you have certain fish that can be used to define the bioconcentration factors or the bioaccumulation factors. To give you an example of what is biouptake and how does it happen, those of you who have studied ecology, you'll be familiar with it. But if you haven't, then this might help in understanding how it happens. So let us say DDT is sprayed on a field. Where is it going? It goes into the soil, it goes on the leaves of the plants that are in the field, it goes into the air, some of it goes into the air, and some of it will end up in the water that is flowing through the fields, the irrigation water. Okay. Now this water is going to end up in a surface water body. From the water body, you will have phytoplankton. These phytoplankton are basically algal cells and aquatic plants. And these algal cells and plants are not eating, they don't eat the way we eat. They are absorbing these compounds from the environment, which is an aquatic environment, through their, literally through the skin. So it's the dermal effect over here. Or you might even call it sorption. These phytoplankton are food for zooplankton. These zooplankton are insects, worms, larvae, anything that you can think of that are feeding on phytoplankton. Now, these are slightly larger organisms. They can be visible to the naked eye or they may not be visible. These zooplankton are then eaten by small fish. You can see an example of a minnow here. These are very small fish. And these fish serve as food for the predatory fish. So you have these large fish, the predatory fish, which feed on the minnows. You also have birds, which you can see in the top left corner. You have this tern family, and these terns are being fed. Literally, the parent terns are feeding their chicks with minnows. So you can see how it is being transferred through the different trophic levels. And then you have the scavengers as well as larger birds. So you have the herring gull and the merganser, which is a fish-eating duck. They are going to feed not only on the small fish, but also Hello?
Hello?
Yes, ma'am, it's
Is
okay.
there any-
It's audible.
No,
No
ma'am, no problem.
No
Yes,
problem.
Yes, no problem. Continue.
Right. Okay, so these scavenger organisms like the gull, which will clean up the dead organisms, and then you have the fish-eating duck. These are large birds, which all thrive on any of these organisms. So as you travel up the up the food chain, what you see is that the numbers of the organisms at each level are reducing, but the biomass of the individual organism is becoming bigger, and it is also feeding on more and more of the lower organisms. So if you think about phytoplankton to zooplankton, what is the ratio? It may be 100 to 1000. And then how many zooplankton are eaten by minnows and how many minnows are eaten by the large fish. So that's how it gets, the compound is being concentrated in the tissue of that particular organism. I'll give you some numbers over here. We'll focus on the lower part of this table. It's from a textbook, and it's from a textbook on environmental science and engineering. And you can see the water concentration. The water concentration is 5 times 10 to the minus 5 ppm. That is our baseline. The concentration factor here, by definition, is 1. We plankton. It has gone from 5 times 10 to the minus 5 ppm to 4 times 10 to the minus 2. And the concentration factor is 800. From plankton to the minnows, the concentration factor, which is what we are interested in, is 18,800. Predatory fish, you have 26,600. Then you have birds that feed on the small fish, you have 78,200. And you can see how as you go up the trophic pyramid or up the food chain, you see the concentration factors rising by orders of magnitude. So this is something that is a huge problem. It not only endangers the of certain species, and I'll give you a few examples of that, but it is also problematic for human beings as well. So I'll show you some examples a little bit later. Now, I've been talking about bioaccumulation, bioconcentration, or biomagnification. This is a biological process. For those of you who are familiar with biology, you know that biology takes a lot of time and effort. If you're doing research in any field of biology, it takes a lot of time and a lot of effort. And even then, because of the variability in nature, the results that we get are generally extremely variable. So it's always helpful to have a chemical surrogate that will help us to understand the biology, so to speak. So here is an example from a early paper, long time back, 1984. A paper was published where you have the bioaccumulation factor versus what is called the octanol water partitioning coefficient. Now, the octanol water partitioning coefficient is the rate of the concentration of that compound, of any compound. You pick any compound, any organic compound, any synthetic organic compound. If you dissolve it in octanol, normal octanol, versus dissolving it in water, you will get different concentrations in the two different solvents. That ratio, the log of that ratio, is what you see on the x-axis. How does it relate to the bioaccumulation factor? So you can see a straight line over here with 41 different chemicals. And you can see how good the regression is. The regression equation and the R-square value for this particular regression is extremely good. It is 0.9. So here we have a biological process that is extremely well correlated to a chemical parameter. You can see that the octanol water partitioning coefficient is telling us something. It's a much easier factor to measure, and you can find it in any textbook. Most of the chemicals are well understood. Most reference textbooks will have these numbers. So when you know the octanol water partitioning coefficient for any particular chemical, you also have a pretty good idea how it will behave in the biological world. So this is an extremely important finding because it tells us another thing, and that is what I want to show you over here. So here, if you see, we have the list of chemicals in the first column. In the second column, we have log KOW followed by water solubility and Henry's constant. These are the three different sets of parameters that are there. Many of you will be familiar with this textbook by Sawyer, McCarty, and Parkin, Chemistry for Environmental Engineering and Science. Let's take our example of DDT. DDT has a log KOW value here of 6.9. So that means that 10 to the power 7, almost 10 to the power 7 times higher concentration of DDT happens in octanol compared to water. You can see the concentration in water is extremely low. This is in milligrams per liter, and the concentration in octanol is very high. So what does that tell us about the bioaccumulation potential or the bioconcentration potential of this compound? That means this compound is likely to accumulate in biological tissue by orders of magnitude, by almost 7 orders of magnitude. So that's an extremely high biological bioconcentration factor. Okay, this is an extreme. I think DDT is one of the extreme ends, but we are commonly using lindane, we are using malathion, parathion. These are all very common compounds that are there in our agricultural practice, and you will find that they all have pretty high log KOW values. So it's not just about DDT. DDT is just a simple example. So here is lindane. Lindane is 3.72. Which was part of the mixture of Agent Orange, 1.78. Now, as long as the value, KOW value, is greater than 1, it means that the compound is hydrophobic. Hydrophobic means it doesn't like water, it prefers organic. If you know, polar likes polar and all of that. So it will prefer to dissolve in alcohol, in this case octanol, but it will also prefer tissue, the organism's tissue, and that means that the bioconcentration potential is very high. So we have other common compounds. I have phenol here, that's circle, DDT. Lindane is something that's common in this area where I stay, and 2,4-D, I gave you an example of that as well. So there are many other compounds that are similarly capable of bioaccumulating in the environment. There are more examples of bioconcentration factors from another textbook. And I need to remind everyone about one more point, and that is the difference between organic compounds and metals. Remember that metals, when they are thrown into the environment, they tend to form complexes with organic compounds that are present. So it may be natural organic matter, which will provide a means for the metals that are discharged along with wastewater. They will form complexes with with the natural organic matter and become more soluble, as well as bioaccumulate in the tissue of organisms. The best example is mercury. You have all heard about the Minamata disease, and you know that that happened because of the bioaccumulation of methyl mercury. Mercury was being thrown out with the wastewater, it got methylated. So the organic form of mercury was bioaccumulating in fish tissue. The fish were being eaten by cats, birds, dogs, human beings, and so on. So it traveled through the food chain. And because it's a neurotoxin, it caused serious problems. So there were fatalities as well as serious illnesses because of methyl mercury accumulation in the trophic chain, trophic pyramid. So you have several other examples over here. So you have arsenic, you have organic compounds like benzene, chlordane, which is a pesticide, chloroform, which is a disinfection byproduct, chromium, which is a metal that also bioaccumulates, but not to the same extent. If you compare it with DDT, which has a bioconcentration factor of 54,000, and you see the metal factors, the metals have very low bioconcentration factors in comparison to compounds like DDT. So there is a huge difference, but that does not mean that toxic heavy metals do not bioaccumulate. They do bioaccumulate, but not to the same extent as organic compounds. I'm going to spend a little more time on DDT because, like I said, it's very well documented. There is a lot of interest in it, and there has been a turnaround in terms of the usage of DDT. So it was invented a long time back in 1874, but the insecticidal properties were described more recently in 1939. And Paul Muller got the Nobel Prize, I think, for this. There was widespread use of this compound during and after World War II as a mosquito repellent. And because it remains the best mosquito repellent, it was really good for controlling malaria and typhus. And that's why there's been a turnaround about the use of DDT, especially in countries where mosquito vector diseases are very common. India is one of them. Africa is also another continent that has major problems with mosquito-related diseases. Let me say a few more things. It was banned in most countries because of the publication of Rachel Carson's book, The Silent Spray. Let's take a look at the half-life of DDT and its two very common intermediates. So DDT is dichlorodiphenyl trichloroethane, and it has two byproducts. This is not ODE, it's actually DDE. You have DDE. DDE is dichlorodiphenyl ethylene, and then you have dichlorodiphenyl dichloroethane. Okay, so these are the two intermediates. There are several pathways by which the two intermediates are formed, and it's also known that these intermediates can be more toxic than the parent compound. DDT is fairly toxic, but the intermediates can be even more toxic. And you can compare the half-life of the same compound under different environmental conditions. So you look at the half-life of DDT in soil, it goes anywhere from 4 to 30 years. In sediment, it's much shorter, 1 to 4 years. In water, it's shorter still, 26 to 56 days. And in the atmosphere, it doesn't stay very long. It's not very volatile. It's a high molecular weight compound, so it's 1.5 to 3 days. Similarly, you can compare the half-lives of the intermediates as well. Now, like I said, these half-lives are very useful for determining the persistence of the compound. And when you understand the persistence, then you also understand the bioaccumulation factors. This is basically what I just said to you about the use of DDT. It was used for agriculture in 1945. It became the best tool for malaria control in 1955. It was prohibited in 1972. And that was by the US. And then by the Stockholm Convention, everyone agreed no use of DDT for agriculture. But then in 2006, the WHO said, for control of malaria, DDT is okay. So with certain restrictions, it has been reintroduced. Structures. This is the parent compound, DDT. Most of it is present in the form of the parent compound. And you have 75 plus 16.5%. This is the mixture of DDT that we buy. And you also have the intermediate. So you have DDD and DDE. What happens to it when it's thrown into the environment? From another source, I have some more data about DDT in water. So if you have 3 times 10 to the minus 6 ppm in water, in zooplankton, it's 4 times 10 to the minus 2. In minnows, it's 0.5, in large fish 2, and in fish-eating birds, ospreys are very large fish-eating birds, it goes up by another order of magnitude. So you have 25 ppm. Just to revise for those of you who may be confused, ppm, it's parts per million. Parts per million in liquids is milligrams per liter. And when we talk about tissue concentration, we measure it in the terms of milligram per kilogram. So it's parts per million, so it's milligram per kilogram of tissue. So milligrams of the compound per kilogram. Of the compound per kilogram of the tissue. So when we think about what are the problems caused by DDT, to summarize some of the problems, the biggest thing was the environmental impact on several species of fish and birds. The word Silent Spring of Rachel Carson's book, the title, it comes from the idea that birds were being endangered mainly because of the thinning of the eggshell. So DDT was accumulating in the tissue of the birds, and when the eggs were laid, the shells were thinner than normal. Birds sat on the eggs to incubate them. Because the eggshell was thinner, the egg broke before producing a chick. So eggshell thinning was one of the major reasons for for several species of birds, the bald eagle, the falcon. This was all in the US. So all these birds were endangered mainly because of the accumulation of compounds like DDT. It was found to be toxic to fish and birds, and it really had an impact on the reproductive potential of these organisms. In terms of human health impacts, it is an endocrine disruptor. So you are all familiar with the idea that hormones determine every activity within the organism. So whether it's a human being or any other organism, especially the higher organisms, hormones are absolutely, they are present in nanograms per liter, you might say, and they determine metabolic activities, they determine reproductive activities, and several other types of activities. So anything that disrupts the hormonal system of the organism is called an endocrine disruptor. And there are several methods, there are several mechanisms by which these compounds disrupt the endocrinal system of organisms. It has been detected, DDT has been detected in human tissue, it has been detected in breast milk, and so on. So, reproductive and developmental defects have often been blamed on the presence of DDT. Leaving DDT behind, let's take a look at some of the environmental factors that determine the persistence of any particular pesticide. So we have a list of the major factors, and let's just go through them very quickly. The first and foremost is sunlight. So anytime you throw anything out in the environment, you know that sunlight is an oxidizing agent and it is responsible for breaking down any material that is basically exposed to sunlight. So it can break down chemical bonds, it can break down products. So you have high molecular weight compounds that are broken down into low molecular weight compounds. Then you have microorganisms. In any environment, especially soil, sediment, water, all these environments have a large number of microorganisms in terms of species as well as in terms of numbers. So bacteria and fungi, which are amongst the lowest level of microorganisms, they are capable of biodegrading any organic compound. Remember that we're dealing with xenobiotics. So you will find that the natural bacteria that are present in any particular environment, they are not ready. They are not immediately ready to degrade a particular new compound, a new xenobiotic. So it takes them a while. So you have a long acclimation period that is required for these bacteria to adjust to the presence of a new compound. But once they do adjust, they will break it down into smaller compounds and eventually they may even mineralize it to plant and animal metabolism. We're all aware of the fact that we eat different types of food and it gets broken down as it passes through our bodies. So that is basically what all plants and animals are also doing. So they are taking in material from the environment. Animals are ingesting organic material and plants may be ingesting CO2 by it's always CO2, but they are also capable of excreting certain compounds into their environment, especially into the root zone, which can also modify the other compounds. Okay, so plants and animals can change chemicals into forms that become more soluble. Remember, we are dealing with hydrophobic compounds. So to make them dissolve better in water is one of the first steps in making them more biodegradable. And this also makes the removal from the body easier. Water is the universal solvent, and like we always talk about that. But in this case, we are dealing with compounds that are not dissolving in water as readily as most other compounds. So water helps to break chemicals apart and it basically is through hydrolysis. It's generally considered to be a slow process, but if compound is hydrophobic, it is not within reach of hydrolysis. So the first step is often making it hydrophobic, making it hydrophilic, and then dissolving it in water, from where on you get more and more biodegradation. Then we have dissociation. Dissociation can happen by any pathways. There are several chemical pathways by which dissociation can happen. So you have large chemical compounds being broken up into smaller pieces. Then you have sorption. Sorption includes two processes, adsorption and absorption. Both of these result in chemicals.
भैया ये क्या है तेरे क्लास चल रही है क्या
हाँ चल रही
Participants may please check, there is somebody's mic very
गाने कभी चलते हैं क्या
Mathur, may please check, Vidit Mathur, if you can hear me. Vidit Mathur, if you can hear me, please. Yes, who's this participant? Vidit Mathur, it seems. May please check your mic. Participants may please check your mic. I can see maybe this is Vidit Mathur. No, that's great. Yeah, madam, please go ahead now. Please continue.
Okay, right. So I was talking about sorption, and sorption can be either absorption or adsorption. In either case, the chemicals will stick tightly to the particles or to any other media that they are exposed to. And because they are sticking to the particles and the surface of these particles, they generally become inaccessible or difficult to degrade or biodegrade and so on. And they also move along with those particles. So if you have a situation where the compound is present, let's say in groundwater, or somehow in the subsurface, it goes into the subsurface, then those are good examples about how these chemicals become inaccessible, or in other cases, they move along with the particles. I'll go into more details later. And I've already said a lot about bioaccumulation. So I have already explained the whole process of bioaccumulation and bioconcentration, so I won't explain that again. We'll move to the next point, and that is the degree of persistence. So if you see, there are some examples here of the half-life of different pesticides that are present in soil. So you can see that something like dicamba, which we don't have in India, has a very long half-life, 30 to 60 days. And we, if you look at all the household pesticides that we are using, they all belong to this group, pyrethrins. So the pyrethrins have a very short half-life, which is a good thing, because if you want something to degrade easily so that it doesn't stick around in the environment for a very long time, then this is the right group of compounds. And you can see the same compound, permethrin, is another example of one of the pyrethrins. And it is present in soil, water, plant surfaces, indoors. And in all cases, depending on temperature, depending on other ways in which it has been applied, it will determine the half-life. So if you see soil, you have 40 days; in water, 19 to 27 hours; on plant surfaces, 1 to 3 weeks; indoors, it can be even more than 20 days. It depends. This is in particular is a very temperature dependent factor. Now I come to the second part of what I would like to talk about, and that is bioremediation of these xenobiotics. So how do we control these xenobiotics? What are the situations in which you might find these xenobiotics, and what can we do about them? So one of the things is that has happened in the past, maybe in India as well, and that is you have accidental or sustained releases of xenobiotics in the environment. I have heard of news reports, and I think a few years ago there was a major oil spill off the coast of Chennai, and there was very often you hear about accidental releases of pesticides into the environment and so on. So even recently there was an incident in Visakhapatnam where you had the release of a particular chemical into the environment. So these kinds of xenobiotics are often released either accidentally or intentionally, where people are not treating the wastewater in which there are xenobiotics. So you have these kinds of releases of xenobiotics into the environment. And you have to control as well as remediate the situation after it has been defined or quantified. Now, when we talk about bioremediation, we normally think in terms of biodegradation. Is the compound biodegradable, and can it be biodegraded under aerobic conditions or anaerobic conditions? So whether oxygen is required or not required. That is the first set of questions. Why do we want to go for biodegradation? I've already shown you so many environmental factors. We have photodegradation, we have absorption, we have chemical reactions, all these things are there. So why go for biodegradation? The answer is very simple. It is cheap. Biodegradation is basically using nature to do nature's job. Okay, so literally engineering the situation to make it. So it's basically a cheap and effective method. That's why I've written cheap and effective method for utilizing microorganisms to degrade xenobiotics. So what you can do is you can expose the microorganisms, the native biota of a particular area that is affected by the compound, by the xenobiotic, the biota, including the microbes, are exposed to that compound. And what is often missing in nature is adequate nutrients. For degradation to happen, especially by microorganisms, adequate nutrients are essential. So by providing time for acclimation and adequate nutrients, you can make this happen without much difficulty. And I will show you some examples of exactly that. And you can go all the way to harmless end products like carbon dioxide and water. So you have, remember that we are dealing with xenobiotics, which are compounds that are present at ppm or lower levels. So when you have complete oxidation, where the end product is CO2 and water, so that's complete oxidation of the compound, and the amount of carbon dioxide generated is not much. So this is, like I said, using nature to do the cleanup. When we think about biodegradation of xenobiotics, we need to think in terms of long-term and short-term effects and concentrations, routes of exposure. I don't think I'll go into any of those details, but we also need to be clear about these few terms. I've been using the word persistent up to this point. So I've already mentioned that persistent means resistant to degradation. And recalcitrant is when a chemical is resistant to biodegradation under all conditions. Now you might say, are there recalcitrant compounds? We use the term very often. In the literature, you'll find that very often we say this is a recalcitrant compound. It's because it's very, very difficult. What we really mean is that it's extremely difficult to biodegrade. And true recalcitrance in nature is not really possible. And we come to a somewhat humorous definition, and that is bacterial infallibility. Bacterial infallibility is literally a principle where you say that you give any compound to a consortium of bacteria and adequate nutrients, adequate environmental conditions, you will find that the bacteria have a way of degrading that compound. So they are infallible in the sense that they can deal with anything that they are exposed to, provided you provide the nutrients and the right environmental conditions. Because when we, I'll just add another point over here, when we think about microbiology, you look at any extreme environment on the planet, you will find that there are bacteria that are capable of surviving even under those extreme conditions. It may be the bottom of the ocean, it may be the ice caps, anywhere, wherever you can think of it, you will find microbes, especially bacteria. So that's why true recalcitrance is not possible. They are capable of doing the job. They just need either nutrients and or the right set of environmental conditions. You can go all the way to mineralization. And this is what we aim for. When you have a xenobiotic in the environment, you want it to be converted to harmless inorganic products that are mineralized. So we call that mineralization. So we have CO2, water, ammonia, sulfate, sulfide, chloride, and so on. Okay. So these are harmless, or even if they are not so harmless, these are mineral end products. And these inorganic end products can no longer be toxic, at least under the normal conditions. Biotransformation, where the organic compound is not completely oxidized. In mineralization, we have complete oxidation. So you have the end product CO2 and water. transformation, if you get only partial oxidation, you will get, we call it biotransformation, because you are generating intermediate organic compounds which may be toxic. They may be more toxic than the parent compound, or they may be less toxic than the parent compound. So this can happen under aerobic conditions, anaerobic conditions. It depends on the nature of the compound. I'm going to give you a simple example. Let's say we have all been to petrol pumps and you know that they have underground storage tanks. So you have these petroleum filled underground storage tanks. And let's say over a period of time, it springs a leak. This leaking material will eventually move into the subsurface and contaminate the groundwater aquifer. Any village or home or residential area or shed or schools or what you, any of these areas that are down gradient from the flow of this, from this particular tank, this leaking underground storage tank, that will, when the water is pumped up, that will be ingested. So there is the possibility of ingestion of contaminated water. Now remember, we are dealing with petroleum compounds, and if petroleum compounds are open to the atmosphere, they are volatile. We all know the smell of petrol, and we know that they are highly volatile compounds, so they can be inhaled as well. So you have two routes of exposure for anyone who's, for the common man, and we have inhalation and ingestion of these compounds. And remember that petrol itself is a mixture of thousands of compounds. So all of those compounds is what we are exposed to if you have a situation like this. Before I go to the next point, I'd like you to see if you can deal with this. What I have here is a table that shows you a combination of electron donors and electron acceptors. So those of you who are familiar with microbiology will be able to understand this. But those who are not familiar, please don't get scared. There is a very simple point to be made over here. The organic compound that is shown here is acetate, and it has been combined with different terminal electron acceptors. The end products in all cases are carbon dioxide and water. The last case, we have carbon dioxide and methane. That's fermentation reaction. I will come to that last. Remember, we're talking about biodegradation mainly by bacteria. So when bacteria are utilizing organic compounds, they need organic compounds to serve as a carbon source, and they also need to use these compounds to derive energy. So there are two things that in fact, all organisms need that. We also eat food for the same reason. We need the carbon, but we also need the energy that comes from the breakdown of the material that we eat. In the case of bacteria, so this is thermodynamics. Let me keep the bacteria out of this. So this is the combination of electron donors. So acetate is the electron donor, and oxygen in this case is the terminal electron acceptor. The value for this reaction is the highest in the sense that it is minus 106 kilojoules per electron per equivalent. And as you go down the list, the second electron donor, I'm sorry, again wrong. The second terminal electron acceptor is Fe3. Fe3 is being converted to Fe2. The third one, nitrate, is being converted to nitrogen. Fourth one, MnO2, which is Mn4, is being converted to Mn2+. Sulfate, in the next case, is being converted to sulfide. And in the last case, we do not have a terminal electron acceptor. Acetate is being split into two compounds. One is more reduced than acetate, and the other is more oxidized compared to acetate. So that's our fermentation reaction. What is the point I'm trying to make here? When you have organic compounds in the subsurface, there may or may not be enough oxygen. If there is oxygen, the bacteria will obviously prefer oxygen. Why? Because the energy yield from the reaction with oxygen is the highest. Everyone wants more energy with the same food, right? So this is the first thing. And then you have terminal electron acceptors, you have iron. Oxygen is not there, but iron is there, they will survive. They have sufficient energy to survive. And as long as the delta G value is negative, that means the bacteria can utilize that energy to reproduce and to survive. If value is positive, the net value is positive, then they cannot survive. They have to find their energy from other sources. So when we talk about primary substrate is what I'm looking at here. So this is acetate serving as a primary substrate. So if I have petroleum compounds similar to acetate, they may be able to serve as primary substrates. Then you have nitrate going to nitrogen. You have Mn4 going to Mn2 and so on. So you can see where the least amount of energy is. We talk about biogas. Biogas is good for us, but not for the bacteria. The bacteria don't get much out of it. Okay, so that's the least. So we have two types of situations. We have aerobic or anaerobic situations. The first one is strictly aerobic in the presence of oxygen. The last one is fermentation. There's no respiration. And I call it fermentation. The bacteria are capable of surviving. We all know that. We know that biogas is generated, but they don't have much energy to derive from this particular combination of compounds. The other terminal electron acceptors have decreasing amounts of energy to be derived. But it's possible for the bacteria to survive under all conditions as long as any one of these terminal electron acceptors are present. Any one of them will do the job. One will do the job more completely, like oxygen will do it far better than any others, but the bacteria are fully capable of surviving and reproducing. So coming back to the idea of a spill, let us say we have an accidental spill into the subsurface. What will happen? As long as there is oxygen, these kinds of compounds can be degraded. As the chemical seeps deeper and deeper into the subsurface, you will get less oxygen. So you have oxygen that is going up and then down because it may be depth dependent or it may be utilization by the compounds. Is there a question? Hello? Okay.
Yeah, yes, ma'am. No problem.
Yeah, go on.
Yeah. No, ma'am, no question. I'm just carry on.
Okay.
Okay,
So
if the
that
participants, they are willing to ask questions, maybe they can, raise a hand, or maybe they can unmute and interrupt the speaker, and that's what Madam is willing.
Sure.
So in between also.
Sure.
Please.
Okay, so I'm continuing. So here we have an aerobic environment where many of these compounds can be degraded, and you can see that oxygen levels, this is the one. This is the one where you have oxygen levels declining immediately. Now that can be by two processes. One is biodegradation, and in this case, you can also have less oxygen because the water is going deeper into the surface. Then we have denitrification, where you have increase in the nitrate levels for some of these, if they have nitrogen and then none of them do. So you have the use of nitrate in the denitrification reactions. And remember, the whole point here is just this, that supposing we take a situation where all these terminal electron acceptors are available, that the bacteria are free to use whatever is available, and all of them are available. then the bacterial consortium is going to gobble up acetate or any other organic compound with the oxygen. So this is going to be fastest as well as the highest energy yielding reaction. Then they will move to the other reactions. Yes, they will move to the other reactions or the other combinations with different terminal electron acceptors. So you have denitrification, you have the use of Mn4 and Fe3. So you have reduction of these compounds, and then you have sulfate reduction, and finally methanogenesis. So these are some of the things that can happen in the subsurface at any contaminated site. I've already mentioned that this example was for a primary substrate. So when I was talking about acetate, or we are talking about some of these compounds, it is mainly based on the understanding that that organic compound, whether it's acetate or any of the organic xenobiotics that we are dealing with, this organic compound can serve as an energy source and or a carbon source. But you can have another situation, and another situation is where the microbes do not get either energy or carbon from the growth. However, they do degrade the compound. And I will show you some examples, at least one example in a little bit. And there is another situation where the compound itself serves as the electron acceptor and it serves as the energy source for that. So the example that I showed you of fermentation, where the same compound is both electron donor as well as electron acceptor. So secondary substrate utilization can happen in three different ways. One is co-metabolism. So if you have a primary substrate like, let's say, glucose or acetate or some of the readily degradable compounds, and along with that you have DDT, trichloroethylene, polychlorinated biphenyls. These are recalcitrant compounds. By themselves, they are fairly recalcitrant, but in the company of a readily biodegradable compound, they may get degraded more easily. Then you have another situation where the compound, the SOC, the xenobiotic, serves as the electron acceptor, and in the process is reduced while the primary substrate is oxidized. So let's say you have a combination of acetate with an SOC. So that is another form of secondary substrate utilization. Then we have a third situation, which is very common, where the xenobiotic concentration is extremely low. Just imagine a situation where you have high amounts of glucose or readily utilizable organic matter like starch or glucose or whatever else it might be. And in that situation, you also have a pesticide where it's at ppm or lower levels. Now, this concentration is too low for the microbes to survive on as a primary substrate. So the microbes will utilize it, but they will have to look for a primary substrate which will allow them to reproduce more easily. Examples of examples of previous case. I have shown example of manganese. So manganese and iron is what I've already seen. There are other examples here of chlorate, selenate, dimethyl sulfoxide, which is very common in seawater, arsenate, which is common in the groundwater over here. We have TMAO and you have fumarate. So any of these are different types of electron acceptors which can also be paired up with xenobiotics for, in this case, anaerobic respiration. Let me give you an example of what I just said about co-metabolism. So here we have toluene. Toluene is a petroleum-based compound. In the presence of oxygen and water, it will be degraded to cis-toluene dihydrodiol. And we have toluene deoxygenase, TDO, is an enzyme that is secreted by the bacteria to break down toluene into cis-toluene dihydrodiol. Now remember, these are aromatic compounds are difficult to degrade because the ring structure is more recalcitrant than the aliphatic structure. So these aromatic compounds, the first thing you need to do is break the ring. And this is exactly what TDO is doing. TDO is breaking the ring and making it an open structure, making it, that's the first and most important step. Now, this TDO is also capable of attacking another compound which may be present in the same environment. So you have trichloroethylene, which is an unsaturated compound. It's a simple unsaturated compound. It will also be attacked by TDO and you get TCE epoxide. So you have co-metabolism of TCE along with toluene in the same environment because the enzyme is there and it doesn't know the difference. It will attack whatever is there. So this is an example of co-metabolism. So let me give you a few general principles. Biotic biodegradability. The first one is molecular structure. I'll show you a good example of that. Now here in this slide, we have three types of polymers. The first one is starch, the second is glycogen, and the third one is cellulose. Starch, as you all know, is part of potatoes, rice, what we eat, those are all common forms of starch. This is a very simple polymer. All it is glucose. In fact, all three polymers that I have shown you over here are all glucose-based polymers. And most of you must be knowing that glucose is the most rapidly biodegradable compound, not just by human beings, but by bacteria as well. So here you have a straight chain polymer of glucose, and the nature of the bond between one glucose monomer and the next monomer is the alpha 1,4 bond. Okay, so it makes a long straight chain, and I think all of you know we when we are feeling tired, we add sugar to our water, make tea, coffee, etc. So that's the big pickup. So that's the simplest way of getting glucose, and that's our energy source. And it serves as an energy source for the bacteria, for plants, for animals, everyone. Drops. Then we come to glycogen. Glycogen is a branched polymer of glucose. So here you have glucose, and at different points you have a different bond. So the straight chain is formed by alpha 1,4 bonds, but the branches are formed by alpha 1,6 bond. So this glycogen is used in the cell wall of different surface receptor molecules. That is one of the examples of a branched polymer of glucose. Then we come to another one which is common around us. If you look at plants, they have a rigid structure. They are able to stand upright against gravity. How do they do that? They are able because of lignocellulosic compounds. And these lignocellulosic compounds are just glucose. Lignin plus glucose. We won't talk about lignin, but here we have a glucose polymer. The bond is the difference. The bond is a beta 1,4 bond. I won't go into any of the details, but that is what gives rigidity to plant and algal cell walls. Okay, so the ability of plants to stand upright against gravity is basically because of these compounds. And even though the basic chemical composition is the same, their biodegradability is extremely different. So let's take a look at it. Starch is straight chain with alpha glycosidic bonds. Glycogen is branched, and so is cellulose is also straight chain, but with a different type of bond. So beta glycosidic bonds. Now you all know that if you're eating potatoes versus some other vegetables which have a large amount of cellulose in them, you know which one is easier to biodegrade. Potatoes are extremely easy. Potatoes and rice, they're pure starch. They are the easiest to degrade. We all know that. We observe that on a daily basis. There are other vegetables which are not so easily biodegraded even by human beings. So you can imagine that bacteria have it even more difficult, right? So starch is, they are all 100% glucose. Starch is the easiest to biodegrade, followed by glycogen and followed by cellulose. Now to functional groups. You have different functional groups over here. So you have aromatic rings. Remember, aromatic rings are more difficult to degrade compared to aliphatic compounds. Then you have these functional groups attached to the different aromatic compounds. So you have toluene, ethylbenzene, xylene at different locations. You have metal compounds at the ortho, meta, and para positions. So you have under aerobic conditions. You have xylene is more degradable than toluene, which is more degradable than benzene and ethylbenzene. In the meta position, is harder to biodegrade compared to ortho and para positions. Those of you who are well versed in organic chemistry, you can probably go through the literature and find out why that is. The bond saturation is another major issue, and we have saturated aliphatics and unsaturated aliphatics. So you have glucose, which is a saturated aliphatic, and you have unsaturated aliphatics like trichloroethylene and so on. So you will find that saturated aliphatics are much easier in general to degrade compared to unsaturated ones. Straight chain is easier to degrade than branched chain. Solubility. Aqueous solubility, I've already explained to some extent. If the compound is in water, it's going to be easier to biodegrade. If it's away from water, water is necessary. It's the necessary media for bacteria, especially bacteria, to survive. And as long as there's water and the compound is hydrophilic, it will be degraded by the bacteria in some period also. But if it's hydrophobic, it becomes difficult for the bacteria to degrade it. I'll show you examples of exactly that. Then we have some more generalizations. Carbohydrates, am I running out of time? Okay.
Yeah, I think it's fine.
Yeah, I do have a few more slides, so I'll try to go through them as quickly as possible. So simple carbohydrates. The best example again is glucose, glucose acetate. They're all simple carbohydrates. Amino acids, they're all highly biodegradable. Fats and oils are a slightly different story. They are slightly different because they have very low aqueous solubility. They are hydrophobic in nature, and that is the factor that makes them difficult to biodegrade. So if you can reduce their hydrophobicity, then it becomes easier to biodegrade. Straight chain saturated aliphatics, which can be biodegraded aerobically, but not anaerobically. So the first step in the pathway requires oxygen, and unless oxygen is present, you cannot start the biological process. Unsaturated aliphatics can be degraded both ways, aerobically as well as anaerobically. There's several other factors that are listed over here. So you have straight chain versus branched chains. You have alcohols versus aldehydes and hydrocarbons. You have primary, secondary carbons and nitrogens versus tertiary and quaternary structures. Then you have aromatics, which may have chlorine or nitrate containing rings. Then you have ortho and para positions versus meta positions on the rings. The number of rings, for example, naphthalene, DDT, they all have more than one ring. So these are all examples of factors that will determine the biodegradability of the compounds. In the beginning, one of the most common environmental problems that we have been looking at in the last few decades is oil films or oil slicks. So these are a real problem because if you have discharge of either wastewater or accidental discharge of oil from boats and ships or from pipelines and so on, what that does is it destroys the environment, especially the ecosystems, because organisms that are present in any ecosystem are not capable of dealing with hydrophobic compounds like oil. So the entire ecosystem gets disturbed, and many of you may remember the Exxon Valdez oil spill, and I'll show you some example of bioremediation of that. So I'll just go there. Now, petroleum compounds can be degraded at different rates. And you can also have methane oxidation. I'm going to avoid that topic. We'll just drop that. So when we talk about bioremediation, there are two ways of doing it. Let's say we have a contaminated site. You can dig up the soil. If the soil is contaminated or the subsurface media is contaminated, you can dig it all up, take it to an industrial facility or a large scale facility, and then bioremediate it. But what we prefer to do, and why bioremediation is successful and popular, is when it's done in situ. So whether it's a subsurface situation or whether it's a surface situation, in either case, in situ bioremediation has been most successful in comparison to other types of processes. What that is, you have to stimulate. Stimulate is to encourage the growth of microorganisms in the subsurface or on the surfaces. Use the natural biota. Whatever is present in that natural environment has to be encouraged to utilize the xenobiotics that have accidentally been released into that environment. So for that, if you want to make it aerobic, aerobic will give you the best results. You have to provide adequate air. So you have to aerate depending on the situation. If it's an aquatic environment, you have to provide air externally. If it is a soil environment and it's exposed to the atmosphere, you won't need aeration. Other thing that you need is nutrients. Very often what you see in these contaminated situations is lack of nutrients. Remember, the bacteria need a large number of nutrients. So we have, you already have carbon, hydrogen, and oxygen, but you also have requirement for nitrogen, sulfur, phosphorus, calcium, magnesium, and the list is extremely long. Now, this can be done under both aerobic as well as anaerobic conditions. So depending on the nature of the contamination, you can deal with either aerobic or anaerobic conditions. To go through this example because I really like it. It's simple and it's highly effective. It's one of the biggest success stories. For those of you, like I said, who remember the Exxon Valdez oil spill, which happened in 1989, thousands of kilometers of coastline were basically contaminated by the oil that was spilled out of this oil tanker. And this was the Alaskan coastline. So you can imagine that the Alaskan coastline, where the temperatures are very low, the entire coastline, it's a very, very sensitive region in terms of its eco-sensitivity. You have a slick, you have a layer of oil on the entire beach. The birds and fish were all covered with oil on the surface, and you can imagine what happens to a bird if it has oil on its feathers. It could not fly. So many of them were dying or unable to survive because of all these problems. So this is the beach contaminated with a surface of oil. Here you see oil droplets. Now you can see that the surface of the droplets is infested with bacteria. So you have hydrocarbon oxidizing bacteria in large numbers on the surface. Why can't they go inside the droplet? They cannot live inside oil. Oil is hydrophobic. Bacteria like water and they are hydrophilic. They cannot deal with hydrophobic environments, and they have clustered at the interface between the water and oil. So you have this interface between oil and water where the bacteria have clustered, and if the nutrients are available, then they can degrade the oil. Here is an example of in situ bioremediation. So you have this beach, the Alaskan beach, which was contaminated, and the contamination resulted in this dark color. So you have this blackish blue-gray color in the photo, and you have this patch which is lighter in color compared to the remaining area. What we did over here was sprayed a mixture of nutrients. Again, those of you who are familiar with the BOD test, you know that we create nutrient media. Media is inorganic mineral nutrients. So these mineral nutrients are added along with, are just basically added to this patch. That is all that was added over here. And the native bacteria were able to degrade the oil. And you can see it's lighter in color because the oil has been degraded over here compared to the other patches which have not been sprayed with the nutrient mixture. We have some more examples. We have a subsurface example over here. So let's say you have groundwater that is contaminated. Again, the same thing is done. The fluid mixture, which contains nutrients along with air, is injected into the subsurface. So let's say we have a petroleum spill in the subsurface, the example of the petroleum leaking tank. So in that case, the nutrients will flow along with the groundwater, and you have another well down gradient from the injection well where you can either recover the water, and one of the benefits of doing this is that if the compounds are hydrophobic, these compounds can be brought into solution and then recovered, or they can be allowed to degrade within the subsurface without recovery. So these are different types of methods of in situ bioremediation. There are a large number of factors that will influence the efficiency of bioremediation, especially in situ bioremediation. So you have microbial consortia. One bacteria may not be able to do the job. It may be a different group of or different groups of bacteria. It may be one species, it may be multiple species. It all depends on the condition. So you have microorganisms or their consortia that will degrade the material. What is the degradation pathways? What is the reaction stoichiometry? How fast is the reaction? What are the electron acceptors, nutrient requirements? Are there inhibitory compounds present in the contaminant mixture? What is the toxicity? Let's say you have something that is extremely inhibitory to the bacteria, like mercury containing compounds, then they can be very toxic. And even though bacteria won't die, all of them won't die, they will still survive, but they will bring down the biodegradation rate enormously. What basically the activity coefficients that we normally think about that, and then we have reactions with soil or aquifer matrix, we have chemical equilibrium constants, we have sorption constants. At the mesoscale, that was all at the microscale. Then you have the mesoscale, you have absorption, which I've already mentioned. It may be at equilibrium or non-equilibrium conditions, depending on how old the contamination is. Then you have the attachment and detachment of microorganisms. Are they sticking to the surface? Are they detaching themselves from the surface? All of that. Do we have methods for quantifying the microorganisms? Is there diffusion in the subsurface? Is there plugging of the porous media, interface transferred from liquid to solid and even to aqueous, to gaseous systems? All of that becomes important at the mesoscale. Then at the micro scale, at the large scale, you want to look at the convective flow of water and water that is flowing. Because remember, what are we concerned about? We are concerned about the quality of water that we get at our tap. If I am dependent on groundwater, I want to know if there is oil in my groundwater. I want to know where it's coming from and whether it's harmful for the health of my family and me and all of that. So these are macro scale factors. So what is the rate at which the groundwater is flowing? Is it dispersing? Is that contaminant dispersing? What is the spatial heterogeneity? What are the hydrologic properties, boundary conditions? All these things will make a difference to the efficiency of bioremediation. I'll show you some examples over here. So you have the initial substrate concentration of various BTEX compounds. So BTEX is short for benzene, toluene, ethylene benzene, ethyl benzene, and xylene. So these are petroleum-based compounds. And you have the degradation rate on the y-axis and the substrate concentration on the x-axis. You also have different electron acceptor conditions. So you have aerobic, denitrifying, and fermentation. So under aerobic conditions, you can see that the degradation rate is always highest. Aerobic means highest energy yield as well as fastest degradation in general. It works if you have three methanogenic points and the rest are denitrific conditions. So the denitrifying rates are not very different from the aerobic reaction rates, but the methanogenic rates are much lower compared to the aerobic or the denitrification rates. In terms of comparison between lab studies and field studies, I think it's always very important to remember that what you see in the lab is not what you're going to see in the field. In the field, in the lab, you have controlled conditions. You're controlling everything and perhaps playing with just one factor. In the field, you have to deal with all kinds of factors, tens of factors, hundreds of factors. So the efficiency that you get or the degradation rate that you get in the field is nothing compared to what you see in the lab. So you can see the lab degradation rates. As the substrate concentration increases, the degradation rate in the lab is showing an increase. But in the field, there's no impact at all. So it's very difficult sometimes to understand why the field conditions are not giving the same results as the lab conditions. But that's because you're dealing with many more factors in the field compared to the lab. In summary, I'm going to stop here. This brings me to the end of this lecture. So the main thing is that xenobiotics are now designed to be easily biodegradable so that you minimize environmental impact. So environmental impacts includes human health impacts. Remember bacterial infallibility. Very easy to remember that if you give the bacteria a chance to acclimate themselves to the presence of these synthetic organic compounds and you provide nutrients to them, they are almost certainly going to degrade the compound. Secondary substrate utilization of these compounds is possible. So they can be biodegraded. You just have to find the right combination. Combination of terminal electron acceptor. So you have T, terminal electron acceptor, and ED, electron donor. The right combination of these two compounds will determine the energy yield. I've given you the example of acetate and the degree to which the electron donor, in our case, very often it's a xenobiotic compound. The degree to which that xenobiotic will be biodegraded will depend on the environmental conditions as well as nutrient availability. You have large-scale contamination, thousands of acres or thousands of kilometers. I've given you the best example of that. One of the simplest examples is the Exxon Valdez oil spill. It's very easy to bioremediate it. It's cheap, it's effective, and it can be done. Required is a little patience. It takes a little more time. It's not chemistry. Chemistry happens almost instantaneously. Processes take a little longer, but they're cheaper and very effective. Results in the field are not always as efficient as in the lab, and that applies to biology as well as chemistry. So in either case, you have to be careful about what you're dealing with in the field, because in the lab, everything is controlled. In the field, you have no control. So designing solutions has to account for all the factors that you're likely to encounter in the field. I will stop at this point. If you have any questions, please ask. I'll try to answer them. Thank you. Thank you for listening.
Thank you, Professor Goel. In fact, it was really wonderful. And this xenobiotics and its adverse impact on environment, was altogether new for me, at least, maybe for many of us. But then it was really, put in very interestingly. We got to know what all are the solutions for this particular problem, and how it affects different kind of environmental ecology. And so it was very learning experience for us. So thank you so much for this nice talk and presentation by you. Yes, now we can take some questions. Yes, there is all appreciation coming your way in the chat box by the participants.
Yeah,
Thank
you, everyone.
Yeah,
Hello,
they're
ma'am.
appreciating a lot. Thank you.
Ma'am, hello.
Yes.
May I ask a question?
Yes, please.
Surely.
Yes, please.
Hello. Yes.
This is Dr. R.C. Chipa, right? If you first identify
Yes,
yourself,
yes.
and then ask a question, please.
Yes, yes. When we have to talk about the various organic compounds which are toxic in nature, so the
Yes,
one thing is their solubility in water.
yes,
The phenol, DDT, whatever, acrolein. So you have mentioned acrolein, phenol, DDT. So acrolein
yes.
have high solubility as compared with the DDT, and DDT has a very little. So
Okay.
the solubility of these organic compounds, how these can be correlates with bioconcentration.
Uh,
In case of the bioconcentration,
yes,
one minute.
that is the crux of the matter. If you think about tissue, whether it's bacteria, algae, human beings, it doesn't matter, plants, all of us are organic in nature. We are organic, we are the sum total of organic compounds, right? We have 70% water, yes, but that water is either in the blood or in other body fluids. We are not concerned about that. The biomass, the mass that we are, or if you look at a plant or you look at any other organism, we are all the same. But basically, we are all the same. So that, excuse me, that biomass is organic in nature. It's made out of various organic materials. You have carbohydrates, proteins, nucleic acids, all these things. Okay. Now, like dissolves like. So you have something like DDT, which is hydrophobic. It, it does not want to be in the blood. It does not want to be in water. It's going to look for tissue. It's going to accumulate in tissue. Okay. So that is the point over here, that this is a hydrophobic compound, and because it's hydrophobic, it does not like water and it will concentrate in the tissue. So whether it's fish, whether it's human beings, whether it's plants, whether it's bacteria, algae, doesn't matter. All are organic material and it will prefer organic material. That's what KOW is all about. You have an organic solvent. Octanol is an organic solvent and water is basically water. And when the KOW value is 10 to the power 6.91, what does that mean? It means that this compound 10 to the power 7, approximately 7 times higher preference for organic solvents compared to water. It does not want to be in water, it wants to be in organic solvents. Where are the organic solvents in the body? Organic solvents are the tissue, the hard tissue and hard and soft tissue, yes. And in many body fluids, which are organic solvents, not water. Have I answered your
Okay.
question?
Yes, yes, ma'am. My another question concerned with the some pollutant or persistent organic pollutants, whatever the health hazards on human, and you have also mentioned about the endocrine disruptors.
Yeah.
So, how these endocrine disruptors correlates with bioconcentration factor?
Different.
The same
They are
thing.
very different. These are two different issues, and they are very different issues. In fact, endocrine disruption happens in many different ways. There are at least two mechanisms in my mind right now. One is that the body does not recognize the difference between a natural hormone and a synthetic organic mimicking. You know, the synthetic organic compound mimics the work of a natural hormone, and the body accepts it without realizing that the synthetic organic compound does not have the properties that will allow the metabolic activities to proceed in the right manner. So it disrupts the hormonal activity of the organism. So that is one mechanism. The second mechanism is where the synthetic organic compound actively destroys the hormone. Okay, so there's a chemical reaction where there's complete destruction of the hormone or the gland. I'm not so sure about that, but those are the two mechanisms that I'm aware of. I think there are some other mechanisms as well, which I'm not aware of, but that does not have to do with the hydrophobic hydrophilic aspect. That's a different matter. Endocrine disruption is a different matter.
One of the disruptive, whatever the concern of endocrine disruptor, and that is the PCB. PCB, dioxin.
PCBs are capable, yes, PCBs are capable of doing both. In fact, DDT, PCBs, many of the pesticides, they're all capable of doing both things. They can disrupt the endocrine system and they can bioaccumulate. So my talk was focusing on the bioaccumulation aspects. I have not said much about the endocrine disruption. But these are two different mechanisms by which these compounds are toxic to not just human beings, but to all organisms.
The last question, last question, whatever the electro accumulation, you have great experience. One of them you have explained. Electro accumulation.
Electro accumulation.
Yes.
I am not aware of that.
With the help of electric field, with the help of conductance, or whatever the applying certain electricity into that. So by which we can create a certain toxic material.
Yes, I think I know about electro-oxidation. Electro-oxidation is when you take the synthetic organic compounds and break them down into simpler forms. That's an engineered process. That's not a natural process. So that's an engineered process, which again I have not talked about. That was not part of what I wanted to talk about. So it's kind of outside the scope of what I was trying to do. But electro-oxidation of synthetic organic compounds is a well-known method of treating and breaking down synthetic organic compounds. Yes.
Thank you. Thank you.
So this is.
Thank you, Dr. R.C. Cheap. I think you got the answers, for a lot of your questions. So thank you for having this curiosity, madam. There is
so
one question.
I just yeah.
One or two questions from the chat.
Dr. Swati from. Sir, this is Dr. Swati from BIT Sindri. I've got one question to put. If you could kindly connect me to ma'am.
Yeah, please ask your question, madam.
I can hear you. I can hear you.
Yes, yeah, please go ahead with the
yes,
question.
Ma'am. Actually, as we all know, microplastic pollution that is emerging as one of the very burning issues regarding as far as the environmental issues are concerned. So, and these microplastics, they are very fine plastics, which are basically entering into our, we can say, precious water resources, especially our beaches. They are very fine plastics, and they are entering inside the aquatic ecosystem. They enter inside the fishes with the help of certain, we can say, with the help of the food chains. They are bioaccumulating in the tissues of the fishes, and they are creating a lot of concern. During the entire presentation, ma'am, you have discussed a number of pathways. For example, biodegradation is there, photodegradation is there, degradation is there. What, according to you, is the most suitable pathway for degradation of the microplastics?
I have some students who are currently working on microplastic degradation. And what we found is that UV radiation, in the natural environment is one of the most potent factors. That is one factor. And, a dry environment is also another factor. Less water results in greater, not biodegradation, but degradation. So that's away from the bio part of things. What you have said about bioaccumulation is very true. And these are some of the things that are well known about microplastics, that photodegradation, chemical degradation, including hydrolysis, all these are happening all the time.
Okay, so thank you, madam. And I suppose, Dr. Swati, you again sent answer. You were very attentive in the first session as well. I appreciate that. So that's good. Right.
Thank you
Right.
so much.
Okay, Professor
Thank you
Goel.
for listening.
Yeah, there's a question from a participant, Mafidur Rahman. Yeah, that's by chance if DDT is sprayed inside a living room, will it be detrimental to human living there?
Definitely, definitely. See, this is one of the biggest concerns right now because in African countries, the death rate or the mortality rate due to malaria and mosquito vectored diseases was very high, and that's why DDT was brought back. And what they have been now using is mosquito nets, which are sprayed with DDT and with they're using wall paint that has some DDT in it and so on to keep the mosquitoes out. There are some studies, I think, that talk about the efficiency of these methods, but it's a difficult topic. And, African countries have been hard hit. And I think in India also there is a lot of positive response to the fact that DDT is the only thing that works against mosquitoes, and mortality rates due to mosquito-based diseases or mosquito-related diseases are very high. So it's a difficult question, but the answer is yes, it's a hazard for human health. But when the choice is between a mosquito-related disease versus mutagenic or carcinogenic potential, I think we are all choosing to avoid acute diseases and deal with chronic diseases, right?
Yeah.
The problem is any mosquito-based diseases are going to be very acute. So whether it's malaria, dengue, chikungunya, whatever it is, it's a very acute problem. It's not a long-term problem, and you want to solve it in the short term. Cancer, on the other hand, cancer, reproductive defects, developmental defects, these are all chronic defects, chronic effects. And I think by and large, there's a global consensus that we avoid the acute effects and deal with the chronic effects. Because, see, the problem is that people don't really have a very high handle on cancer and related defects. Okay. We have much better control over mosquito-related diseases, at least in terms of treatment. So I think it's a very complex issue, but what I have understood is that there's a global consensus towards avoiding acute effects.
Thank you, madam. Thank you. In fact, it was really, really impressive the way you responded to all the very queries by the participants. And I think we all stand educated on this particular new topic. And those of us, maybe in the participants who are from this area, maybe they can appreciate it better. But even the ones, like me, who are, absolutely new to what you discussed today, even we go to learn lots of things. Now we can appreciate xenobiotics maybe better now. So thank you, Madam Goel, for this really, really nice and informative talk, I should say. Thank you so very much, and thank you to all the participants.
Thank you.
And I go on to Dr. Jakar, to say a few words on, on the concluding words. Dr. Jakar, please.
Excuse me, sir.
Yes, please, who's that?
This is Amita from Bangalore. I have one question. I just want to know from them.
Yes, Amita.
One small question. Actually, my research domain in water-related issues, I'm working on water purification processes. So my question is, DDT is not soluble in water, right? So can it kill some microorganism, or can it be beneficial to remove some sort of impurity from water? Because it will be easy after the treatment to remove it from water. Can you give some? Yes, ma'am.
No, I see, because it's poorly soluble. We have an echo over here. It's a very poorly soluble compound in water.
Yes,
so, yeah, so I don't think an aqueous process would be beneficial in any form or manner. Where you would want to use it, because if you're thinking of killing microbes in water using DDT, if that's where you're trying to go, it's perhaps not a good idea, because the compound itself is so toxic to human beings and other organisms.
Thank you. Thank you. I think Dr. Amita,
Yes.
you can answer,
Yes, yes.
right?
Sorry
Okay.
to disturb. Whatever the question is there, drinking water, treatment of drinking water, microorganisms are removed with the help of certain chemicals. Whatever those chemicals may be chlorine, chlorinated water,
Ozone,
chlorine
yes,
oxide,
yes,
and economically bleaching powder is used.
yes.
Bleaching powder itself gives certain adverse effect, but recently, whatever the known, certain pesticides which are used for treatment of drinking water. So those are soluble in water, but those have also adverse effect. But what we have, certainly the microorganisms are so there. So in Jaipur, whatever they are using, that in near future it may be give adverse effect in India. So
Okay.
certain pesticides, certain are used chemicals. Okay, sir.
Yeah,
Thank you.
Thank you, Dr. Chiba. Yes, Dr. Goel.
yeah, I would say that it's not a good idea to use a pesticide for drinking water treatment, because once we know that, see, there is a simple conventional wisdom, you might call it, that if a compound is toxic to any organism, it's not going to be benevolent to other organisms, right? If it's toxic across the board. So you may be killing the cockroach and the mosquito and all of that, but there is a price to pay as well. So we definitely don't want to be using our pesticides for drinking water treatment.
Yeah, right. You're right. Thank you, Dr. Goel. And there's, I think you can have this as the last question in the
But
chat.
whatever the thing is there, so 1 minute. Whatever the thing is there, because the major supply is surface water, and surface water contains so many microorganisms, and it is very difficult to minimize microorganisms with the help of bleaching powder. So bleaching powder itself supplies the chlorine, and chlorine kills the microorganism. But difficult in the during in context, surface water itself is more supplied. So here surface water is more supplied. So that's why we have to use certain pesticides. Whatever the both sides are there. Okay.
Yeah, but then that's right, because the technicality behind Dr. Goel expressed very nicely. I mean, pesticides anyway, if you do something, there's an
We
adverse
don't
effect of that anyways.
want
Dr.
to
Goel.
call ourselves pests,
Yeah.
right?
That's right.
Do we want to treat ourselves the way we treat our cockroaches?
Yeah, very true, very true.
Hello, madam, sir. I have one question.
Who's that?
Madam.
Who's that? Who's that?
Actually, which one enzyme is the most appropriate one for the bioremediation of this DDT type of this pesticide?
It's any one enzyme. At least I'm not familiar with any particular enzyme that would be the answer. But I do know that there are several studies in the literature where you can take a look, and there are bacterial consortia that create certain enzymes for degrading DDT. So that has been done in soil environments, in aqueous environments. All this kind of work has been done for a long period of time. So I don't have a specific enzyme in mind, but I do realize that there are probably multiple enzymes that are used for degrading DDT.
Okay, ma'am. Actually, I'm working with some of the enzymes that that can be done for the wastewater treatment. So means like if the pesticide is added, then again means like I'm also searching which one will be the appropriate one. So as while doing my work, then it will be a little bit easy. So I was thinking. Okay, thank you,
Yeah,
ma'am.
you might want to look for a review paper that does some, that provides some information in that matter.
Okay, thank you, ma'am.
For you, yeah. You're welcome.
Okay, okay, thank you. Okay, no problem, ma'am.
Thank you, ma'am. And I think this participant seemed to be from northeastern part somewhere. And who is that? I don't know. I mean, you need to identify first, and then that also can appreciate where you come. So it's okay. Thank you for the interaction. All the listen to. And really, thank you so much, Madam Goel. In fact, it was very, very informative and very nice talk. Thank you so much. And Dr. Jafar, please conclude, with your comments on.
Thank you, sir. Thank you. On behalf of organizer, thank you so much, madam, for your very nice and very informative presentation and session. And I think your session is very excellent and wonderful, and all have enjoyed, especially on genobiotics and the history of DDTs and their harmful effect and remedies, biodegradation, and all. And as especially we know the in canal irrigated areas where the use of pesticides, chemical and fertilizers, the effect every family has at least one cancer patient, maybe two or three. And ma'am, one train is running from the Sri Ganganagar to Bikaner, and where hundreds of patients are coming every day, and its name is known as a cancer train. So that effect, everyone knows the effect of these chemicals on water as well as in irrigation. So it's a very nice, informative talk and very wonderful. Thank you so much, ma'am, and thank you all participants for your good interaction and for your patience. Thank you. Thank you. Over to Professor Sato.
And thank you for inviting me and for listening. Much appreciate your attention.
Thank you so much, Dr. Goel, and thank you, Dr. Jakad. I think with that, it was really very good talk and very interesting to hear you. Thank you so much, madam. I think now we can close the session, and Dr. Goel will say a document with your email. You can do the needful revert back. Thank you. So thank you all. Thank you so much.
So shall I stop sharing and...
You can stop sharing your screen, ma'am, now. That's
Thank you, ma'am.
That's right. Now you could be on full screen also, in case. So बहुत अच्छा लगा, ma'am. I think it was a learning experience, personally. And we'll connect you maybe in future as well to NIT Kurukshetra or maybe otherwise for some other academic activities and interactions. Right.
Sure,
So thank you so much.
sure.
It was really nice, really
Thank
nice.
you. Thank you. Right.
Thank you,
And
sir,
I
and
think
thank
the
you to all the
meeting-
participants.
Yeah, please.
Now end the meetings.
Yeah, you can now close this session, Ravi. No problem. And Madam
Okay,
we'll share the document. You please do the needful and revert, right?
Right. Thank you. See you. Bye.